Ladies and gentlemen, thank you for tuning in once again to Live with Andy. I'm your host, Andy Dybala. I appreciate you taking a break out of watching Bravo. I know when you search Live with Andy on YouTube, it goes to Live with Andy Cohen. You know, all the Bravo birds just yipping and yapping at each other all day, and Andy Cohen tries to keep them under control. So I appreciate you guys taking a break from your normal programming and tuning into my show, Live with Andy. It's more of a research-based, citizen journalistic type show. So without further ado, I guess give me a follow on Twitter at Live with Andy as well. Make sure, a lot of people kind of wonder, like, Andy, post your content in the chat. Make sure you post it, link it up on YouTube after the show. But mainly, if you follow me on Twitter, you'll get all the links I talk about on the show here. And we know Elon Musk has taken over. He's making it more of a free speech platform. So it would behoove you as a free speech patriot to go to the Twitter platform and sign up right now. I know a lot of you guys were banned earlier. Just make another email, sign up. It's pretty easy, and follow at Live with Andy, and you can get all of my research updates in between the shows. One of the things here, as you can see, there's 17 million minks were culled in Denmark because of COVID, and recently in Ohio there was 10,000 minks that broke out. It's actually near my hometown of Toledo, Ohio. There's someone that broke into one of these mink places, and they stole, and I guess all the minks broke out. So if you're living in that area, make sure you get tested for the COVID-19 vaccine, because that is the reason that they killed 17 million minks in Denmark at the beginning of the pandemic. So that kind of leads me into what we're going to be getting in here today, and that's genetically engineered food, and that goes to how our food is vaccinated, to it being genetically modified itself, to its DNA being CRISPRed and tinkered with by scientists in the laboratory. We're going to be digging into a whole host of subject, or a whole array of genetically engineered food subjects on this show this evening. I do appreciate all you guys tuning in. Thank you for clicking that email that I continue to send out on neighborhood news, and for all of you guys just participating in being patriots and not giving into the deep state mandates that are plaguing our planet right now. But let me say hello to the chat here before I dig in. We got Deborah Johnson. Bring it on. She loves caveman science. It has been a while since I've done caveman science. There's been a couple people that have mentioned it. So I was like, all right, let's dig back into the science aspect of things and make it a little less political. Corky Goss is in the chat. As you can see, I got my Cleveland Browns hat on. I was having a bad hair day this evening, so I had to throw a Browns hat on. We got a big game this weekend. It's getting snowed out in Buffalo, so they're moving the game to Detroit. I guess it's record cold temperatures for this time of year, even though it's global warming and we can't play football outside anymore. We got Tyrone Sargent. He's saying hello in the chat from Pittsburgh. Tyrone recently sent me some very eye-opening research that they're doing in the University of Pittsburgh relating to like Planned Parenthood and like some of the things they do with aborted baby fetuses and scientific research. And I think we're going to be digging into that next week as he settles back into the cold state of Pittsburgh for the weekend. We got Amber Connor, Caveman. What's up, Amber? Mr. Green Jeans. That's two of the Protect the Nest 3 ospreys that are flocking around the internet nowadays, Mr. Green Jeans and Ambicon. Make sure you check out Protect the Nest on McDuff. They do it every once in a while. It's a great show, and they dig into how to protect the family. Greg Clark, what's up, Greg? Lynn Williams, how's it going? Go Browns! Hell yeah! Your Atlanta Falcons beat my Browns. Amber Connor. We got Tyrone Sargent. Browns won't lose this one. We'll see. I beg to differ. I'm a Browns fan. I don't have a positive outlook when it comes to football games on Sunday when the brown and orange are on the field. So at the beginning of the show, I just showed you guys the minks and on my Twitter account how they broke out in Ohio, and that kind of incorporates this whole One Health approach that's been going on this in this plane. Where'd it go? You think my leeway would be a little bit better here? So when it comes to genetically engineered food, um, this One Health approach is at the um, forefront of the science and the technology behind it. And when it comes to vaccinating animals, what they're vaccinating them with, how they're genetically modifying the animals, genetically engineering them, the release in mosquitoes into the wild. You know, Bill Gates is working on this called gene drive therapy, where they're genetically engineering the mosquitoes, sending them out into the wild, and they're repopulating the genetically engineered mosquitoes are populating with the natural mosquitoes, and their gene is the dominant gene, and it takes over eventually. And hopefully, in the future, there will be no more malaria. And the One Health approach is how wild animals and our agricultural animals like pigs and cows and stuff, they interact with each other. You know, the bats in the cave that bat lady was working on, they fly over the pigs and they poop on the pigs, and the pigs, you know, they get infected with the bat diseases. Then us as humans come along, kill the pigs, whatever, eat them, and we get infected as well. It's this whole One Health approach in trying to limit the spread of diseases. And this has kind of been a new thing within the past decade of our global government. You see it in all the health documents. They always talk about One Health and what it's about. And I do have a little video here for those of you who have never seen it before, just breaking it down real quick. So here we go. This is by the U.S. Department of Health and Human Services and the CDC. One Health, from concept to action. Walking around, you know, we're all going to be living in cities, and we got tribes in Africa. We're going to be interacting with each other. You know, the cities like interacting with people in the smaller communities. This is where we get our livestock from. You know, we got these big farms with all the pigs and the cows. Get the milk. We get our meat from them in the big cities, and it's all a big interactive One Health approach. This video is actually quite slow, and I'm gonna stop it right there because it is boring the crap out of me, and I can't take that elevator music anymore. So you kind of get the point with the One Health approach. They're trying to get a grasp on diseases and it spreading from the animal population to the human population. That's the overall One Health approach. And one of the things they figured out in the early 2000s is that, like, before we vaccinated animals. Yes, we do vaccinate animals. We were giving them antibiotics, and yes, this a lot of people may know this, but antibiotics in your food, what's causing the rise in antibiotic resistant bacteria in our food supply, and why you should buy antibiotic free food. As the use of antibiotics in farming and raising livestock has increased, new antibiotic resistant bacteria or super bugs are emerging. Here's what you need to know about antibiotics in your food. So, pretty much, they were pumping the cows, they were pumping the chickens, they were pumping the pigs with antibiotics. And us humans, we would, along the process, they would kill the pig, process them, and we would eat them. We would eat the animals. And we were actually getting the antibiotics as well in our system, which was making us more susceptible to some of these diseases. So there's this big push in the early 2000s. Here's another article by the CDC. Antibiotic resistance linking human and animal health, and you can tell here thus the One Health. I don't know what just happened right there, but they dig into the One Health approach and how we need to limit the use of antibiotics in agriculture. And I'll get to how this links to SARS in the future of this show here, so bear with me for a moment. So it's actually was a huge issue in the World Health Organization. They wrote an article about it. They got 17. This one's titled $17 billion spent researching new drugs in the past 5 years could be lost just as quickly without global action. And if you dig into this article. So, over the past 5 years, more than $17 billion has been spent by the pharmaceutical industry on research and development of medicines used to treat infectious diseases. Unless drug resistance is tackled quickly, much of that investment could be lost. Today, WHO is launching a comprehensive strategy to contain the spread of drug resistance. This carefully structured framework can be used by anyone involved in the use or management of medical treatments, from patients, doctors, blah, 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 the veterinarians. This strategy is designed to promote the wiser use of drugs so that the resistance is minimized and effective treatments can continue to be used for generations to come. And they dig into antibiotics here. Use of antibiotics in food production also contributes to increased drug resistance. Currently, 50% of all antibiotic production is used in agriculture, not only to treat sick animals, but also to promote livestock and poultry growth. Drug-resistant microbes in animals can be transferred to humans. To prevent this, the global strategy recommends a series of actions, including obligatory prescriptions for all antibiotic use for disease control in animals and phasing out antibiotics as growth promoters. Drug resistance is a natural blah, blah, blah. I just kind of told you what happens. They give the animals antibiotics and the humans get it as well. So this is a big issue when it comes to farming and agriculture. And they had to start developing new methods and new ways of controlling diseases within the swine, within the cows. And they started actually vaccinating the animals. But as you can tell here, there's definitely some issues when it comes to some of the research. I got some different articles here. Here's UNC research uncovers evidence of human susceptibility to emerging coronavirus strain in swine. So one of the things with coronavirus, SARS, was the bats in the caves. We got Ralph Barrick here. He did his research with UNC, and he's the one who actually authored this article. We got Ralph Barrick working on re-engineering different coronavirus strains that don't actually exist in the wild. He's creating them out of nowhere in his lab just in case one day these bats, they're intermingling with each other in the cave. and they're creating new coronavirus strains. They fly out of the cave, they poop on some of the pig farms in China or Ukraine or other places in the world, and you know how it goes. The humans get infected from that point on. And this is a huge deal, and this is actually what Ralph Baric and Bat Lady were working on when it comes to SARS. And you can go back onto my Live with Andy Dybala, my backup channel, and I have a video of Ralph Baric synthetically reverse engineering SARS, him just going through all the science himself. So you can get it from the horse's mouth, not just me talking. Just go to my channel, and it's him reverse engineering SARS. It's actually a great watch if you've never seen it before, and you'll sit there and be like, they created SARS in a lab out of nothing, and it's and this is the purpose of that research, to limit our agricultural herds from getting I don't know what just happened. Our herds from getting sick and getting us humans sick. So before the actual breakout of SARS in China happened, there was a SADS breakout, and they were actually taking all these pigs and throwing them. I don't know what happened to my video. I hate Twitter sometimes. Videos that went across the internet where they were pretty much hurting all the pigs. They were digging holes. They were throwing like bulldozing them into the holes, and then they were throwing thermite on them and setting them on fire. And this was before the actual SARS breakout happened in Wuhan in 2019. This was in early 2018, and here's video proof, and they're going to bury the pigs alive, set them on fire. I'm not going to get into the noise because it's quite horrific. And here's proof that it actually happened. Same thing happened in Ukraine too. We got these United States proxy biolabs that we set up in different countries. When we had some in China, we had some in Ukraine, and lo and behold, there's some swine flu breakouts. We got 364 Ukrainians died from swine flu, and this was in 2016, before the actual pandemic broke out as well. So this big One Health approach when it comes to limiting SADS and SARS and coronavirus in animals has been a huge project amongst these scientists in the health industry for a while, because as our population is growing, we're going to be moving into the cities. Our farms are getting bigger, and there's going to be some more diseases spreading in these farms. I live here in Texas now. I don't know if you've ever driven by some of these cow pastures. It smells like a disease. To say that it is not a pleasant thing for your nose to be driving down the country road with the windows down in Texas sometimes, because it's an unbearable stench that hits the nose, and it literally smells like SARS is being created in some of these farms that you drive around. So I do have some videos here. So I showed you guys that they're turning away from antibiotics in meat because it's affecting us humans. It's affecting our health and this One Health approach. So they got to dump it. It's like, no, this is not allowed anymore. Like, if we're all going to get sick from dumping antibiotics in our meat, then like, what's the point of it? Let's come up with a different approach. So as you guys know, with SARS coming out for us humans, they had this new mRNA vaccine that they used for us. This new DNA technology. But before they gave it to us as humans, they definitely gave it to our farm animals, our friendly farm animals. And the main company that invented this technology was Merck, and it's actually called Sequivity. New and evolving diseases and pathogens represent a continuing threat to the food animal population. Sequivity, a revolutionary RNA particle technology from Merck Animal Health, is at the forefront of vaccine technology designed to combat this ongoing risk. So before they started giving you guys, us, the human population, mRNA vaccines, they started giving our animal population mRNA vaccines as well. And I just showed you how antibiotics affected us humans, not, it affected us poorly. A lot of us were getting sick because the diseases, they got used to the antibiotics in our system, and they developed beyond those borders. So what happens when we're giving our animals mRNA vaccines? And if you're giving our animals mRNA coronavirus vaccines, is this the start of SARS? Is this how it initially, we can't figure out? We can't seem to pinpoint the start of SARS, can we? Is it because we were giving our animals vaccines? We're starting in Wuhan, Brazil, the United States, all around the world, we're starting to give these animals mRNA vaccines. And then us humans are coming along in our grocery stores and eating the food. Is this the spread of coronavirus? It's just a question I'm throwing out there. But here's a little video so you guys can get a break from my lovely voice and just digging into what Sequivity is.
Influenza A virus in swine is an infectious pathogen that causes frequent outbreaks of respiratory disease and is prevalent in swine worldwide. Influenza transmission occurs through nose-to-nose contact and the air. The subsequent pneumonia results in reduction of growth rate and feed efficiency, and an increase in secondary infections and medication costs, which all have a negative financial impact on the pork industry. The swine influenza A virus has 8 segments of RNA in its genome encoding 10 proteins, including viral hemagglutinin, or HA, the most abundant envelope protein and an antigenic focus for vaccination efforts. The use of traditional influenza vaccines helps to prevent disease spread. However, influenza viruses are constantly evolving through mutations and reassortments, causing large strain diversity among swine herds and ultimately poor vaccine control through lack of cross-protection among strains. Merck Animal Health offers an innovative and flexible RNA particle technology, Sequivity, to create safe and customized veterinary prescription vaccines to more rapidly control emerging influenza viruses. Sequivity utilizes an alpha virus-based vaccine platform technology, inserting genes encoding proteins such as hemagglutinin, HA, into the platform to create a vaccine specific to that influenza strain. Due to HA diversity in the field, HA sequences are gathered from clinical animals and analyzed to build customized multivalent vaccines.
Just pay attention, guys. A lot of this is like, that's exactly what the mRNA vaccines are that they gave to us. They swab the pig's nose, they collect samples. There's different kind of coronavirus breakouts in different regions across the world. And they get the samples in their lab, and they go, oh, they're having the HQU2 breakout. And they go, let's give them the vaccine. Let's run it through our computer sequencer and give them the vaccine for that strain. Oh, over here in Brazil, they're having the Cas9 CRISPR breakout. Doing nose swabs, send the sample back to the lab, then they send them a different type of mRNA vaccine to the herd in that area. Now, doesn't that seem kind of like what happened with the human population as well when it comes to mRNA vaccines? All this stuff was used on, all this technology was developed with the animal population before they brought it to us, pretty much. And just pay attention to the similarities here.
While also considering clinical signs and farm history. Merck Animal Health offers geospatial analysis to provide strain diversity information to help veterinarians select swine influenza strains when prescribing custom vaccines. The custom RNA particle vaccine induces a specific humoral and cell-mediated immune response to the HA of the current problematic swine influenza strains. Merck Animal Health is committed to monitoring for new and emerging swine influenza A strains and to empowering veterinarians to proactively update their flu vaccination program with its innovative Sequivity technology. Please reach out to your Merck representative for more information.
All right, now I do have a Merck representative on the a little interview from an expo here, and he breaks it down a little bit more, and just kind of I got just some research for you guys to kind of take in tonight. I got some videos after this and genetically engineered animals and stuff. We're just trying to have a little fun Friday night show here, a little fun biology class on Friday night before our big football weekend. So here is a representative from Merck talking about this new Sequivity technology that they're using on the animal population. And again, some of the things he talks about, just you can kind of replace swine with humans, and it's almost like the same exact thing.
Hi, this is Sarah Mikesell with pigsite.com, and today we're here with Joe Roeder. He is the Director of Technical Services with Merck Animal Health. So thanks for being with us today, Joe.
You're more than welcome.
We're here at World Pork Expo, and you just made a big announcement. Can you share with us a little bit about that?
Sure thing, Sarah. Merck Animal Health, we're very excited that we just launched Sequivity Technology at the World Pork Expo. Sequivity Technology is a novel new approach to creating very customized vaccines for swine producers and veterinarians. It is actually a platform technology that allows us to, in a very innovative new way, create RNA particles for vaccines that are targeted to the specific diseases that swine veterinarians and producers are having a problem with at this point in time.
How does the platform function?
Sure, great question. What we do is in working with the local veterinarian and the producer, we identify the diseases or challenges that they're having. We'll get diagnostic samples, send those to the diagnostic lab, and then identify what we think the causative organisms are. And then we actually get the gene sequences, the genetic information for those particular organisms, and we can then collect those, and we actually have an informatics portion of this as well as a production portion. And then we can actually look and see which are the more prevalent strains of a particular disease-causing organism that we find in that particular herd or that particular area. And then what should we build this vaccine to? And again, this is part of this is just us knowing what's out there today and what.
Again, keep in mind, just plug what he's saying with take away the herds. Like, just think of it as a human population, like plug and play. Like, they're doing DNA modification, DNA editing. From the nasal swabs, they get your gene sequence, and then they can adapt the vaccine to whatever herd in whatever area. Just keep that in mind.
But what is coming, and so it gives us an opportunity to be a little more proactive than reactive, and this is exciting for us, in that it allows us to produce a vaccine that particular farm or group of pigs really needs.
Very good. It sounds like a very customized approach.
Absolutely, it's very customized, and so
he thinks we're all a bunch of pigs.
Sometimes in
lining up for the slaughterhouse
areas or the farms that are having the most problems, we have many different strains that
squealing on Twitter that you guys are editing our DNA.
We're trying to figure out which ones to use, and that's where it becomes a very the thing that I really like about it, this provides us a tool to work with the producer and their veterinarian and really figure out what are their goals, what are their objectives, what should we do, how can we build a better mousetrap to serve that farm and those pigs to really get a handle on this disease.
Very good. And I guess, from a very bottom line
we all fell into the mousetrap, ladies and gentlemen.
Producer standpoint, what does this really mean for them?
Sure. That for the producers, well, first of all, if they're having if they're completely satisfied with their current vaccination protocol, then Sequivity technology is probably not necessary. But for those that they think they're having challenges and they need to do something different, flu is a great example. Flu mutates, that virus mutates and changes. In the human side, we see a different flu vaccine every year. With Sequivity technology, we're actually launching here at World Pork a regional flu project where we create the most updated flu vaccines based on the strains that we find for specific regions throughout the United States. And we're going to update that twice a year. So every 6 months, we create the most relevant, updated flu vaccine for that particular region in the United States. So we're very excited about that tool, and it just allows producers and veterinarians to maybe just do a better job at managing that. And again, we're just excited about bringing a better mousetrap to the game.
Very good. Well, thank you so much for all the information today.
We all fell into the mousetrap, ladies and gentlemen. The Merck mRNA mousetrap. Did you sign up to get your genes edited? So this program actually did. Specialized vaccine program starts pigs off right. Custom vaccination isn't eradication, but a positive, proactive approach. April 4th, 2019. So right before the SARS outbreak happens, I showed you that clip earlier. Not a clip, but a little. The man who reverse engineered SARS-CoV-2 himself, Ralph Baric. He was working on different SADS strains, coronavirus strains with swine. This is literally what they were doing in Wuhan with Bat Lady, trying to protect our agricultural animals. This was their job. And right before the pandemic happens, in 2019, what do? They start giving our animals mRNA vaccines. Now, some of these could have been coronaviruses. Some of these were for swine flu. We're trying to pinpoint where did this random disease come from? It came from the wet market. It came from the Wuhan Games. It came from AIDS. I don't know. Did it come from vaccinating our pigs in our agricultural animal herds? Did it come from vaccinating them in early April 2019? So I'm going to read this article here. For a swine veterinarian, no news usually equates to good news from customers and can often mean that the preventative vaccine approach he or she decided to take on farm is working. Oop, I think I skipped. The way I look at things is a vaccine basically sensitizes the body's immune system to the disease antigen, where there can be a bacteria or a virus, says Pat Graham, a veterinarian in Central Illinois who oversees about 25,000 sows. As an antibiotic is just a tool to dip the infection battle so the immune system can take hold and dominate it to get rid of clinical signs. Without the immune system, an antibiotic is really of no value, he says. With a change in veterinary feed directive requirements and pains, I showed you on the beginning of the show, we can't use antibiotics in farming anymore because it hurts the human's immune system. And paying specific attention to antibiotics in general, one of the items Graham has tried to incorporate into his practice is the use of custom vaccines. Now, this is this Merck technology that that I just showed you the videos for. Even though viral antigens, of course, cannot be treated with antibiotics, we certainly see viral infections that predispose sows and pigs as secondary bacterial invaders that can become significant. For this reason, we try to address the virus first and then the treatment of bacterial diseases after that, Graham says. Well, Graham says farms can implement extensive biosecurity protocol, blah, blah, blah. I don't think I'm going to read this whole thing. When there's plenty of air moving through a farm, you have less signs of flu. It is just natural because all that virus is just pulled out of the farm and sent on its way. Where when you get into cooler weather, air movement is decreased. Those viruses just hang around and expose more pigs to higher levels of virus. Graham says he often fields this question, are we going to get new strains into our farm? His response is yes, and that is one reason why he started working with Channing Sebo, a technical service veterinarian with Merck Animal Health, on regional flu virus tracking. This way he could see not only how his farm strains compare to everybody else's, but also what his close neighbors may have that may be different. Sebo exclusively works with Merck's Sequivity and RNA Particle Vaccine technology. She describes the technology as plug and play, where you don't need a whole bacteria or virus to start the vaccine process, but instead can make custom vaccines electronically with sequences. I just played the videos breaking down for you what the Sequivity RNA particle technology is. But there you go. Right before the pandemic happens, we have a farm in Illinois that gets approved to start using mRNA vaccines. We had breakouts in Washington, Chicago, New York, and I believe Houston were like the main 4 hot spots in the United States of the SARS breakout. And it's like, they were definitely working on some of this technology in China. You know, that's where it's a little bit more unregulated than it is here in the United States. That's why we have biological programs over in Wuhan. That's why we had them in Ukraine, Kazakhstan, some of these surrounding countries as well, just because we can kind of do more extreme experiments there. And that's where some of these vaccines were being developed. Like I just showed you the pigs being pushed into the pit. They were set on fire and stuff. I didn't show you that one because it's disgusting. But it was like, is that like herds in China testing out this vaccine? They had the same problem in Ukraine. People got the swine flu. Were they testing out this vaccine over there as well? I think I'm going to be doing a little bit more investigating as we go here, and I hope to find out soon. And I believe it was John Wilkins who earlier said that Merck, what is that? In my opinion, Merck is worse than Moderna, Pfizer, and Amgen put together. Now, we know that Moderna, you know, it stands for mRNA, Moderna. mRNA, that's like the play on words the company uses within its own title. They're the inventors of the mRNA vaccine, and you have here in early May 2018. Guess who invested on an mRNA cancer vaccine partnership with Moderna? That is Merck. There was more money than this that eventually passed hands, but this was like the start of the partnership. So your Moderna mRNA vaccine definitely went into the swine population there in Illinois, and it was tested out in some of these U.S. Department of Defense biolabs around the world. So just kind of trying to put the feelers out there that maybe us vaccinating our herds cause the spread of this disease. I'm not 100% sure. I'm just throwing that one out there. And I am gonna, excuse me, I do need a drink of water. I am gonna switch gears here a little bit. I'm going to stick to genetically engineered food as pig pimpin'. Who said that? I saw someone that just said pig pimpin'. That's what I should rename the title of the show. Pig pimpin', spending cheese. Swell. What do pigs eat? I forget. I'm not a farm person, as you guys can tell. Everyone's like making fun of my farm lingo and the good pasture, blah blah blah. It doesn't smell. Sorry, I'm not Amish, guys. I grew up in a city, not a smart city, as you know, because I am dumb. But I grew up in a city. So cut me some slack every once in a while. So switching gears here a little bit, I'm going to continue down the genetically engineered trail here. But one of the main reasons why I think why we need to be vaccinated, why the pigs need to be vaccinated, why they're working on this, is because of this. Eventually, one day, scientists, they would like to use animals to grow human human kidneys, human hearts, just different human organs. Kill that animal, take the kidney out, and transplant it into humans. That's one of the goals here. The future of science is human to animal organ transplants, and for the very first time, it did happen in New York this past year, and I have a little video explaining the first ever animal to human organ transplant. And video warning, it's graphic.
In the first successful surgery of its kind, a 57-year-old man received a heart transplant from a genetically modified pig. It's not the first time researchers have used pigs' organs. In fact, pig valves have been used in human hearts for many years. In September 2021, surgeons at a different institution successfully attached pig kidneys to a brain-dead patient. Why pigs?
Many good reasons we've selected the pig as the potential source of those organs, mainly because of the size, the ease of maintaining them inside a clean pig facility, the reproductive capacity. The organs are very similar anatomically and physiologically to humans.
This kind of animal-to-human surgery, known as xenotransplantation, is the result of decades of research. Even in human-to-human transplants, without proper treatment, the body will reject the new organ. The immune system identifies the transplanted organ as foreign and targets it with killer T cells and antibodies made by B cells. Over time, these damage the organ and may cause organ failure. To prevent this, transplant patients take medication that suppresses their immune systems. In the recent case using a pig's heart, not only did the recipient take a new kind of immunosuppressive medication, but the organ supplier was genetically modified for a better chance of acceptance by the human body.
The treatment we use to prevent rejection of human organ is not very effective against preventing rejection of a pig organ. So we're using some new immunosuppressive drugs. This is the first time in transplantation we've had the opportunity to manipulate the donor rather than just suppress the recipient.
Scientists modified 10 genes. They inactivated 4 genes, including one that causes a strong rejection response in humans, and added 6 human genes to the pig's genome to make its organs more likely to be accepted by the human immune system. Researchers are hoping that modified pig organs could bring relief.
See, that's kind of a point with all this research, all this One Health approach. It's going to be all-encompassing here in the future.
To people on the wait list. Today, many never get the chance to have an operation. Of the over 100,000 people in the United States, and counting, 17 people die every day waiting for these life-saving surgeries.
Even if we did 40,000 transplants this last year, there's still new patients added to the list every year. There's always an organ shortage, so not everybody will be a candidate for transplant.
So who does get the few available organs? To receive any kind of transplant,
Rich people.
animal or human, patients must meet certain criteria.
Every patient will have a similar kind of checklist to make sure that they are good candidates for transplant. Does the patient has caregiver support? Or are they complying with medications? Do they show up to their appointments? Can they afford the medications?
Ideally, everyone would have equal access to these life-saving treatments, but that's not always the case.
Who is having access to organs, and is the access equitable? Black people, Latinx people, and Indigenous people are less often referred to organ transplantation. They are also less often recipients of organ transplant. And so what we really want to make sure is that we don't limit who has access to this kind of care, this kind of life-altering care.
But xenotransplantation
It's not a race issue. It's a class issue on who gets an organ transplants.
still has a long way to go before it's considered life-altering care. The recent surgeries were highly experimental, a last resort for dying.
By the way, it's never really a race issue. It's always a class issue. That's very hard for me to say with my speech impediment.
Or brain-dead patients. And we don't know how long the transplanted pig's heart will continue to function.
People who are open to xenotransplantation are usually at the end of their life. They usually have been given some diagnosis that is terminal, or maybe they are not eligible for a human organ. And so if they are open to xenotransplantation, we really have to think about, are we preying on people who are very desperate, who will do anything to survive?
The next step for xenotransplantation is to study the heart and kidney transplants in the context of clinical trials.
Xenotransplantation right now still needs to be studied a lot more. We still need a lot more clinical trials. We still need just a lot more information about its success among humans. And so what I want to make sure is that we don't start promoting xenotransplantation without the proper research and giving people hope where maybe it's giving them false hope.
So imagine one day you go to the hospital, and you're like, I'm having some type of organ failure. Oh, I need a new heart. I'm having a heart attack. You get rushed to the hospital and you're just like, why does it smell so bad along the way? It smells like a farm. And you're like, why is a hospital in the middle of a farm? So you go in, they go into the operating room and they're just like, hey, you need a new heart or you're going to die. But luckily, you see that Merck animal farm? I'm in a Merck hospital. You see that Merck animal farm? We got a pig heart for you, buddy. And oink, oink, it's your lucky day. Oh, I said the joke wrong. It's supposed to say it's your lucky day instead of wink, wink. It's supposed to be oink, oink. Wah, wah, wah. I messed my joke up. You guys get the point, though. Yeah, imagine one day you go to the hospital and there's just like a giant pig farm where they're harvesting human organs inside the pigs and they're transplanting them into humans. It's a scary thought, but someone said in the chat there that they had to hear that the pig had to be alive at the time of the transplant. That would be even scary if you're laying there on the hospital bed and you look over and Suey is sitting there looking at you, just petrified. So with the improvement of science, there are some scary Frankensteinish things going on in our future. What's going on, everyone in the chat? Everyone's saying hello to everyone. Norman's in there. We got John Wilkins, brother Dave. I don't know who Dave. We got Dawn. All of the issues are class issues. Yes, they are. So just, kind of putting it out there that you got to you guys that you've with the antibiotics and the vaccinated meat, you've been eating genetically altered meat, and that doesn't even scratch the surface about some of the things I'm going to be getting into next. That's just vaccines. That's just jabs. That's just mRNA vaccines. Some of our future meat out there, some of our future farms, a lot of the animals are going to be CRISPRed, and it's it's unfortunate, but yeah, I got a little video here. So, but it's our future, like why sit here and spend all this money vaccinating herds when you can just delete the genes or whatever that are more susceptible to getting sick. You got stronger, bigger, faster cows out here. But yeah, here's a little video saying how CRISPR technology could change the future.
Meet Genzel, a 2-month-old Angus calf that could signal the dawn of a new era. Gene-edited animals designed by humans for human consumption.
First in the world.
First in the world.
The company that made her says she's born with traits spliced into her DNA that should allow a cold weather cow to thrive here in the Brazilian tropics.
And what we're seeing now is that it works.
You're about to find out why, if indeed this works, our world is about to change, and why the economic incentives driving gene editing technology are so powerful.
Yes, these are not kosher animals, Linda Hole.
With 200 million people, Brazil is a vast country with a vast appetite for beef. That's because the US is the top producer of Angus meat. Most of Brazil's meat comes from zebu.
Do you think the regulators here are open to gene-edited meats?
I think so, yeah. The agricultural sector is very open and keen of new technologies.
Fernando Flores is one of the lead scientists at the Brazilian government's agricultural research entity, Embrapa. He shows us why Brazil wants to up its game.
All right, I'm not going to show you that whole video there, but you get the point. They developed gene-edited cows for human consumption. And lo and behold, they're talking about, oh, in Brazil, people would be up for eating gene-edited cows. But guess what? Earlier this year, in March 2023, the FDA, the United States, the FDA approves first CRISPR cows for beef. So not only are getting your mRNA vaccinated beef and swine, they're already genetically engineered from the start too. So we saw how the antibiotics affected the human population when it came to putting antibiotics in our meat. How is consuming all of this going to affect us in the future? And scientists act like they know, but yeah, I think it could have some sad effects on the human population. As you can see, there's been a lot of like myocarditis, a lot of brain aneurysms, just health issues going on since the vaccine got pushed on us. So imagine just continuing eating this meat, and they got CRISPRed plants as well, plants and meat, like everything we're consuming on a daily basis is going to be genetically engineered at some point in the future. This is why, we got to get away from consumerism a little bit and get back to our own, like, just family, local farms and start going that route a little bit more. But yeah, March 23rd, 2022, the FDA approves first CRISPR cows for beef. The genome-edited cattle were bred to endure climate change, and their offspring will be used in meat production. So yeah, they got this like different type of fur where they can withstand different temperatures. Earlier this month, the FDA approved genome-edited cattle for use in meat production. They were bred with climate change in mind. And they have extremely slick, short hair, which is said to help the animals cope with hot weather more effectively. So you're eating gene-edited meat, not for like diseases, but for climate change. Unbelievable. The cattle breed known as PRLR Slick was developed using genome editing technique called CRISPR, which is used to breed animals with specific traits. Unlike genetically modified organisms, which typically involve adding genetic material from other organisms and result in a plant or animal that would not exist in nature, gene editing utilizes genes already native in a species, resulting in an organism that could theoretically occur through a natural breeding process. So the one they like shit on GMOs, genetically modified, that would never exist in nature, but ours theoretically could. Big difference. This is why the FDA approved it, pretty much. The federal agency called the decision to introduce the beef cattle to be raised for meat low risk after determining that the intentional genomic alteration of the cattle does not cause any safety concerns. Pending a forthcoming safety review, the meat could land on shelves in as little as 2 years. So coming your way, you got your genetically, your CRISPR meat. You know, you're going to have your Beyond meat. You're going to have your lab grown meat. You're going to have your CRISPR meat. What are you going to be eating? Salt Bay. What are you going to be sprinkling your salt on? In a press release, the FDA explained that IGAs are alterations made using molecular technologies that introduce changes to the genome of an animal. According to Successful Farming, a precision breeding company called Axologen in Minnesota is responsible for utilizing the CRISPR technique to produce the slick coat cattle. It's not the first, so if you live in Minnesota specifically, watch out. It's not the first to use the technology on cows. However, in 2020, researchers at UC Davis used CRISPR technology to breed a cow named Cosmo, designed to produce 75% male offspring. So, yeah, the FDA is like, they're approving RNA vaccinated pigs. They're improving CRISPR'd cows. It's going to be on your plate in the future, ladies and gentlemen. Just to let you know, it's not just cows. It's pigs as well. This was in December 14th, 2020. FDA approves first of its kind intentional genomic alteration in line of domestic pigs for both human food and potential therapeutic uses. Alteration intended to eliminate alpha-gal sugar on surface of pig cells. Today, the U.S. FDA approved a first-of-its-kind intentional genomic alteration, IGA, in a line of domestic pigs referred to as gal-safe pigs, which may be used for food or human therapeutics. This is the first IGA in an animal so these are, these other pigs that are going to be next to your Merck hospital. This is the first IGA in an animal that the FDA has approved for both human food consumption as a source for potential therapeutic uses. The IGA in gal-safe pigs is intended to eliminate alpha-gal sugar on the surface of the pig cells. People with alpha-gal syndrome may have mild to severe allergic reactions to alpha-gal sugar found in red meat. So yeah, there you have the FDA. They're approving pigs that are CRISPR. They're improving intentionally genomically altered pigs. They got CRISPR cows in Africa. And what else are they doing, ladies and gentlemen? What else are they working on? Not only is your meat affected. They just spoke about UC Davis. The greens on it, they say you want it like a colorful plate. That's what's considered a healthy meal. You got your greens, your orange, your red, your white, purple, blue, whatever. colorful plate is better for your health in general. Just keep that in mind. So grow and eat your own vaccines is the title of this article, and it's September 16th, 2021. The future of vaccines may look more like eating a salad than getting a shot in the arm. UC Riverside scientists are studying whether they can turn edible plants like lettuce into mRNA vaccine factories. Messenger RNA or mRNA technology used in COVID-19 vaccines works by teaching our cells to recognize and protect us against infectious diseases. One of the challenges with this new technology is that it must be kept cold to maintain stability during transport and storage. If this new project is successful, plant-based mRNA vaccines, which can be eaten, could overcome this challenge with the ability to be stored at room temperature. So imagine you go to your Merck hospital next to the Merck animal farm. Like, all right, you ready to get your jab? You're like, oh, you're just sitting there, you're like sweating. If you're like me, you're sweating. Like, I hate needles. I'm going to pass out. Call the doctor, even though I'm in the hospital. Call the doctor. I don't want the needle. They're like, not so fast, my friend. You know, they just literally like, swipe your credit card right here. And you got to swipe your credit card, and the refrigerator door opens. And on each different level of the fridge is like broccoli. It's your flu broccoli vaccine. You got carrots that are making you immune to COVID. You know, if you got rabies, you eat a Brussels sprout. Whatever it is. You know, that actually, I might back vaccine technology if they get that. I might eat all. I might just give them all to me. Give me all the vaccines. I'm eating a salad, doctor. Imagine mistakes that could happen, you know. You come home, your husband's a doctor, and you just, oh, why is all these vegetables on the counter? And you eat a salad. Next thing you know, you got 19 diseases in your body battling each other with your immune system at the same time. Our future is bleak. As a human race, people, our future is bleak. So yeah, not only is your beef mRNA vaccinated, but your vegetables are mRNA vaccinated as well. Let's hope the Cleveland Browns eat their vegetables this weekend so we can get a W. So I've been showcasing cows. I've been showcasing pigs, but that actually was not the first thing to be genetically modified for human and approved for human consumption. If I were to do a Twitter poll right now and go like, hey guys, take a guess. Oh, someone already guessed it. Mr. Green Jeans. Go fishing, she says. The animal that was first approved for human consumption that was genetically modified was, in fact, salmon.
On the edge of Newfoundland, in the spectacular wilds of the North Atlantic, I'm about to wrestle with one of the great scientific controversies of our time.
Oh, she's a fighter.
That's going to be good, but we're going to catch it too.
Oh, nearly. Oh, they got a bit of muscle. Got him. So that's your pride and joy.
Absolutely. Great fish, isn't it?
How does it feel? Oh!
Well, it was a good fish.
I got plenty of growth hormone and muscle hormone as well.
If you don't recognize the guy with the white hair, he was the third grandpa in Grumpier Old Men. You know how they were always trying to catch the big fish? Well, he caught the big fish. He didn't just catch it. He grew it in the lab. You continue.
Away, right? The story you'll tell at home.
Exactly.
Oh,
you might be asking yourself, can you actually create high-quality live stream?
These are a new breed of superfish, genetically modified salmon, and they're about to make history. They're set to become the first genetically modified animals approved for human consumption anywhere in the world. And that makes their creator, Dr. Garth Fletcher, a very proud man.
How does it feel to create a superfish?
Well, it's very nice. I'm glad you think it as a superfish. It's just a regular salmon, except it grows faster.
A lot faster, up to 10 times faster than normal salmon, thanks to growth genes from other fish species spliced into their DNA.
Now, the key to all of this are the big fish and the little fish are the same age.
They're exactly the same age. Fertilized at the same time and cultured at the same under the same conditions.
All these fish are just 6 months old. The bigger ones carry Dr. Fletcher's
They should call them little Dybalas. That was like my childhood growing up. Like, oh, those two people are the same age. Why is that one huge compared to all the other kids? I might change the name of my channel from Live with Andy to Live with Genetically Modified Andy.
controversial genetic modification. The smaller ones don't.
Commercially, I can see why you want to do this.
Oh, yes. Oh, yes.
But while the fish farmers are hailing this genetic tinkering as a miracle that will feed the masses, environmentalists have labelled these salmon Frankenfish.
Never mind calling you names or the fish names. The thing is it safe to eat? That's the question, not the label of it.
And is it safe to eat?
I think it is. I know it is.
I was going to pick you up on that. Do you think or do you know?
I know it is.
Sometimes you just...
That sounds like a man. A man who hasn't eaten the fish yet. Do you think or do you know? I know it is. How do you know that you just created them? What are the long-term effects, buddy? So they're like every aspect of our lives at this point, are being genetically altered. Everything we eat, everything, every vaccine we do, they're just trying to hit up our DNA. Hey, DNA, we need to make a change. We got a problem here, and we don't like who you really are. We need to make some improvements. All these scientists, I like to compare them to my ex-girlfriends. They start dating you, and they're like, I don't like this guy, and I need to make some changes. So I'm gonna start comparing all these DNA modification scientists. So I'm gonna just put them on the same level as my ex-girlfriend. Put them behind me. Don't think about them anymore. Genetically Modified Andy. There you go. That's the name. Oh, that should be the name of my new show. Genetically Modified Andy. Live with Genetically Modified Andy. So, just after proving the point that you are no longer human and you are genetically altered in every single way, from the food you eat to the medicine you take to the air you breathe, I'm going to end you on this one. What they're ultimately going to do, once they find the perfect genetically modified Andy, what are they going to do? Like, that's the pinnacle of science. When you come up with the perfect genetically modified Andy, how can you improve? I'll tell you how you improve, ladies and gentlemen. You clone. You take that genetically modified Andy.
Meet Zhong and Hua Hua.
And you make more. Now, this has happened already to monkeys, to primates, our close compatriots as humans, monkeys. When they come up with the perfect GMO Andy, it's over for the rest of you guys. I hate to break it to you. Put a cap on it now. Just call it a day.
Meet Zhong and Hua Hua. These twin long-tailed crab-eating macaques are being hailed as a scientific breakthrough. This laboratory in China has succeeded where other researchers have failed. Scientists at the Institute of Neuroscience in Shanghai created the first cloned monkeys using a technique called somatic cell nuclear transfer. The cloning process began with a monkey egg and a monkey fetus cell. The genetically modified process in the laboratory then develops into an embryo, which was then implanted in a monkey. Eventually, the babies were born. What's novel about this process is the nuclei were transferred from fetal cells rather than adult ones. That's different from the world's first cloned animal, Dolly the sheep, which was created from only adult cells. China is the first to successfully clone primates using this method. However, scientists are criticized for pushing ethical boundaries. The Chinese process took 127 eggs to produce 2 monkeys. Animal rights activists oppose medical research on monkeys.
And I should point out that people may argue that it's not ethical to use monkey. There is a Parkinson's disease model in monkey, which has been used quite effective, but it needs to use a large number of monkeys. In the United States alone, they are importing 40,000 monkeys each year by drug company.
Other research.
So the United States is we're just take it. Hey, you got monkeys on your continent? Send them over. We got some lab research we need to do. And we're all sitting there looking at China, Mr. Mooming Poo. And we're like, hey, how dare you clone monkeys? And yet we do all sorts of just unthinkable things to these animals, these intelligent animals. So he's like, how dare you? I'm cloning them.
Researchers are celebrating how cloning primates could help preserve species on the brink of extinction.
The other use is, many primates are on the border of extinction. Gorillas, chimpanzees, many other primate species are under pressure. Cloning may give you a way to maintain the species.
Given the progress in cloning primates, will people be next?
We will never try, and we have I don't think there's anybody are willing to do human cloning, and the society will not permit it. But like any new technology, once it's appeared, there's always possibility of misuse.
For now, China is celebrating the breakthrough. The newborns are just 6 and 8 weeks old. It's hoped that they will lead long and healthy lives. Hannah Hoxter, Al Jazeera.
There you have it, ladies and gentlemen. The scientists. They're the ones, growing up, I always thought the scientists and the doctors, I always thought they were on my side. After doing a lot of this research, I figured out they're on the Nazi side. Isn't all of this just a giant Nazi dream? Just DNA modification, trying to create the perfect human race. What are we funding here in the United States, ladies and gentlemen? What are your tax dollars going to? Not only are they going to Ukraine, your hard-earned money, not only are they going to FTX and then back to the Democrats' pockets, they're going to all this crazy Frankenstein scientific biotechnology research. And how do we capitalize off of this on the back end? When I go to the hospital, the hospital bill is just like, it's out of the room. You ever if you ever ask, like, hey, can you give me an itemized list of everything that you billed me for in a hospital? It's like, first of all, they never bring it because they don't have that much paper in stock. But it's like, the list is a miles long. They just dock you for everything. So if all my tax money is going to the scientific research, this One Health approach for the betterment of humanity, can I at least get a discount? Can I at least keep some money in my pocket if I have some health issues? Please. The Nazi dream. That's what all this DNA technology is. CRISPR, mRNA vaccine, vaccines, that was Nazi thoughts, Nazi technology. That all came from them, all their doctors. Rockefeller and the Nazis Koch's postulates. I believe Koch was a Nazi himself, you know. You got to fulfill Koch's postulates if you want a vaccine. You know, everything, you know, going to space, spying on everyone, creating a genetically modified Andy. It's all a Nazi dream. When God made Andy, he broke the mold. One of a kind, totally original. Calling out science on its face. Yeah. Caveman science. You know, like on the spectrum of intelligent and retarded, I lean more towards retarded. But I'm trying to take these scientists down. I'm trying. One episode of Live with Andy at a time. All right, ladies and gentlemen, that's Caveman Science for this Friday night. I hope you all have a wonderful weekend. As you can see here, go Browns. Ooh, 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 ooh. We're getting close to the big game. Ohio State versus Michigan is coming up soon. Go Buckeyes. You know, there's a lot of, it may be getting dark outside, it may be getting cold, but there's a lot of good football that needs to be played. That's where my mind will be this weekend. Also, make sure you follow Live with Andy Dybala, my backup channel. After talking about genetically modified stuff, my show all day today, I'm most likely going to get taken down. Let me showcase it for you guys here. So make sure you go to Live with Andy Dybala and give me a follow. Make sure you subscribe. Make sure you share it with your friends. I do have. I think I Hey Siri'd something. Did you guys hear that Hey Siri? That was odd. But they're always listening, people. They're always listening. But go to my backup channel, Live with Andy Dybala, in parentheses, backup channel. I am going to be posting a lot of shorts as we go here. I spoke about the Ralph Barrick synthetic creation of SARS and the coronavirus. I posted that video here. I got some Klaus Schwab stuff, some breaking into the Capitol. You know, I have a lot of research, and not a lot of people have time to watch the long shows. So I'm going to be like the little videos that I had here on my channel for this show tonight. I'm going to be posting on my backup channel, so make sure you go and subscribe there. But everyone, I'm getting hungry. The stomach is growling. It's like 20 degrees outside, and I got, come here, Zeke. Zeke, come here. I got, come here. I got to take this little guy. I know you guys love my dog so much. We've been talking about genetically modified animals, like farm animals. Guess what one of the first genetically modified species was? Ladies and gentlemen, guess what? Dun, dun, dun. It was a dog. So here's little Zeke's making his appearance on national YouTube channel. But yeah, humans, we've been tinkering with DNA for a long time, and they're just taking it to the next steps. So everyone, have a wonderful night. Contact me at livewithandy@gmail.com. Follow me on Twitter at livewithandy. Hit me up, give me a call. And everyone, just have a wonderful weekend. Have a little skip in your step when you walk around. Put a smile on your face. When you see someone, go, wave and go, hey, how's it going? You're looking good, bud. Even if they look like shit. Even if they're having the worst day ever. You know? Hey, how you doing, bud? You're looking good. Keep up the good work. You know, it's a little compassion for our fellow patriots out there. Even if they're not a patriot, just a little compassion may change someone's day.